Assalamu alaikum, dear students. Let's have a look on the dihybrid cross. You can see on your slide the example of the dihybrid cross with the Punnett scale. So here is the one individual, male or could be female. Of course, if it is the male, the other one would be the female. It is having yellow and round phenotype, and both of the gene having dominant allele for these genes, capital Y, capital Y, capital R, capital R. Y stand for yellow and R stand for round. Similarly, the second individual, that is green and wrinkled, it is having both of the alleles of both of the genes in recessive form. Okay, so these are the parents or P generation or parents one. So they are crossed and we get an heterozygous individual. So as this is the case of the complete dominance, so both dominant allele they will express capital Y as well as capital R, and the phenotype would be the dominant parent, that is the round and yellow. Okay, so when we intercross these individuals, so they will generate, as you can see on your screen, four type of alleles, or that we call the gametes. So these four type of gametes, capital Y, capital R, small y, capital R, capital Y, small r, small y, small r. So there is one formula 2 to raise power n to determine how many gametes could be formed from the given combination of the genes. So in this case there are two genes. So 2 is the base, its power is n. So 2 to raise power 2, and the answer would be 4. So we can see we have 4 type of gametes. Similarly for the other individual there would be same 4 type of alleles. So now you can see in the next generation they will combine together. So once the sperm and egg in case of human or animal, or here if the both of them, the anther and ovary, they combine together, so they will form the genotype, and that of course is going to determine the phenotype. So capital Y, capital R is combined with the capital Y, capital R, and we have capital Y, capital Y, capital R, capital. Similarly, in the next one, that the result is the capital Y capital Y small y, capital R small r. In the next one, capital Y, capital Y, capital R small r. In the last one, that is the capital Y, capital Y, small r, and this will go on for all of the uh, combinations shown here with the both type of the gametes from the both of the parents. Okay, so you can calculate one type of phenotype, that is basically round and yellow. So we count there are 9. Similarly, the other type would be the green and round. In that case, there are 3 individuals. So 9, 3. Now, yellow and wrinkled. So this one in the black circle, you can see there are three individuals. And finally, the green and wrinkled. So that's only one individual. So though, if you sum together all of them, there would be 16. So 16 different type of genotypes, and they produce 4 different type of phenotypes. So 9 one type of phenotype, 3 another type, and 3 another, and 1 another. So classical ratio for the dihybrid cross is 9 ratio 3 ratio 3 ratio 1. Okay, now this gives rise to the principle of the independent assortment. The, these dihybrid crosses, they educate us about the principle of the independent assortment, that is the Mendel's third law. So allele at a different loci separate independently. So loci is basically the localization or position of the allele on the chromosome. Allele at a different loci separate independently. Okay. Number two, two alleles of a locus separate when gametes are formed of one another. So that we have seen in the last example. Independent assortment is really an extension of the principle of the segregation, because the alleles segregate here, they also segregate, but they segregate independently. Now, the diagram on the right side of your slide, you can see, okay, so there are two chromosomes, chromosomal pairs, and you can see one chromosome is having capital B and small b, and this is basically one gene which have two allelic form, capital B and small b. Similarly, the other, that is capital E and small e. So when in meiosis or during the cell division, each one of them is separated. So here you can see the combination is capital B, small e, then the combination capital B, small e, and similarly capital small b, small e, and small b, capital E. So all possible 4 combinations are there, but each of them they have a different combination. So that means that basically determines that each allele is going to transfer independently. That means capital E is not going to determine if it will combine with the capital B or small b. It can combine with any one of them, but just small. It can combine with the capital B or it can combine with the small b. Similarly, capital B is not going to determine whether it is going to combine with the small e or capital E, and vice versa. So that clearly indicates that each allele is going to, or each pair of gene and their respective alleles, they assort basically independent to each other. But this is possible with certain conditions. When these each pair of homologous chromosomes they separate independent of each other pair in anaphase of the meiosis. But this will happen only when they are not linked. And when they are linked, you can see in this slide, the photograph, genes that happen to be located on the same chromosome will travel together. So they are linked basically. And in this case, the highlighted red diagonal you can see, red diagram you can see, there are two genes, gene 1 and gene 2. So they are too close to each other. So they are basically linked together, and they are located on the same chromatid, and they will always travel together. It means they will not sort independently. But this is not true in case of the alleles, which are either located on different chromosomes or they are located on the same chromosome but with quite a distance between them. So these are not linked. So they can assort independently. especially which are located on the same chromosome, but they have distance between them. In that case, what happens? There is a probability of crossing over, and that crossing over it can generate a different combination in the gametes. So that's why they are sort independent. Now, just like the monohybrid, we have the test cross for dihybrid, and obviously we are if we want to clarify ourselves whether the given phenotype it has underlying genotype of heterozygosity or not. That can be determined with the help of the test cross. Here in this case, you can see the round and yellow, the dominant, and then the genotype, the genotype for this is the capital R, capital Y, small y, and this is heterozygous individuals, and we want to determine whether that is heterozygous or not in fact. For the wrinkled eye green, the small r and small y, the genotype. So they will produce four type of gametes, but in case of wrinkled and green, it will produce only one type of gamete. And when these combine together, so they will give rise to the genotypes in the offsprings. So the first genotype is round and yellow, and round and yellow mean uh, it is basically heterozygote. The second is the round and green. So this is heterozygous. Round means heterozygous for round, but homozygous recessive for green. Uh, Similarly, wrinkled and yellow. That means recessive for uh, recessive for the roundness, and that will yield the uh, wrinkled, and dominate for the color, that is the yellow, and the last one you can see. Now the most important thing is that all of these they have ratio of 1 to 1 1 1, so each one of them is 25%. So this is classical ratio. As we have seen in the monohybrid test process, we have the ratio of 1 to 1, and here we have ratio of 1 1 1. So there are four type of phenotypes, and the ratio among them is the 1 1 1 1, and that determines that the individual which was we were going we are interested to test, that was in fact heterozygotes, not homozygotes. In case of homozygotes, again we will get just one type of phenotype, and that would be that of the dominant individuals. So please stay home and stay blessed. Thank you very much.